Hayes will inform us of the TM activities on the Breast Cancer Committee.
Thanks, Lee. It's a pleasure to be here, and it's a little daunting to give this talk after the initial two talks in the previous talk, but we'll do our best. We've been working very hard over the last decade or more trying to translate correlative science and translational medicine into our treatments for breast cancer. I want to talk a little bit about philosophy first and then go into some of the things we're doing. I don't have time to cover everything, but I'll do my best. I want to start out with very real declarations of conflict. I'm going to be talking a little bit about this so-called cell search assay, and I receive laboratory and clinical research funding from Veridex, and I have a provisional patent regarding its use, but I'm actually not going to show you any of those data today. I want to start out with a committee of the Institute of Medicine in which I had the privilege to serve. It was formed to look at the Duke fiasco that I suspect most of you know. It was chaired by Gil Lohman from our own institution. But instead of becoming policemen, which we had no interest in doing, we really assessed how could the gene expression array that was used to direct patients within a clinical trial have gotten that far without better oversight. And I think probably the most important thing we did, at least in my opinion, was generate this slide, which has become, in my opinion, what every translational medicine and every clinical physician who does oncology should be aware of, and that is sort of the roadmap to go from the far left, which is you've got a good idea and you've generated some data, to the middle slide, which is here, and that is that you then go talk to someone like Chris and say, make a test out of this. I've got a biomarker, I need a test. And actually did a very nice job reviewing the fact that oftentimes what we as clinicians think must be an analytically validated test because it's being used, in fact, has a lot of pros and cons that we don't fully understand. There's some real nuances there. But once we have a test that meets all the criteria that he laid out, there's this bright line, and that is, should we use this to actually take care of patients? And that bright line on the right side can be reached in one of three ways. Either doing what we call prospective retrospective studies using already existing archive specimens that came out of clinical trials. And if that sounds familiar, that's exactly what this group and other groups do better than probably anybody in the world. If we don't have those archive specimens for a variety of reasons, either we have them and they're not the kind of quality we'd like, as the questions I was asking you, Chris, or we don't have them, then we're forced to do prospective trials to validate the ideas we have, and those can either be prospective trials of prognosis, where you're not using it to direct patient management, or more commonly, prospective clinical trials where the test actually directs the treatment. And in the breast group, we have tried to do all three of these in order to move forward. Whoops. And in fact, this is sort of the model that we put this forward for the last site visit, but it's a model we've been trying to use for quite some period of time, and that is to generate clinical trials with clinical data and specimen availability on the left-hand side of the graph, and then make observations that we could then develop assays and biological data and either validate those using our archive specimens or take them back into new trial designs in order to really validate them. And I want to give you some examples of that. Our flagship study is right now is the so-called responder trial. This grew out of the question we had, is the 21 gene recurrence score, the so-called Oncotype DX, predictive for chemotherapy effect in node-positive patients? From the NSABP data, which I'm not going to show, we had substantial and really high levels of evidence that the 21 gene recurrence score is prognostic for node-negative ER-positive patients. But the question we had is, does it actually also predict resistance? Do low recurrence or so-called luminal A patients, are they less likely to respond to chemotherapy even if their prognosis is worse by virtue of being node positive? I think all of you know S8814, Kathy Albane's landmark study in which postmenopausal node-positive, ER-positive women were randomly assigned to tamoxifen or chemotherapy plus tamoxifen, one arm being chemotherapy with tamoxifen followed by tamoxifen, and the other chemotherapy sequentially followed by tamoxifen, published in The Lancet a few years ago, and this really established that chemotherapy was effective, is effective in ER-positive postmenopausal women, and these are the main results showing that the sequential arm, if I can get this thing to work, CAF followed by TAM did better than the concurrent arm, but both did better than TAM alone, but not by very much. So the question is, can we identify why it wasn't better in everybody? We had tissues archived, formalin fixed paraffin embedded tissues archived. We're going to drop out the middle group because of the concurrent interaction and look at the group who got chemotherapy followed by TAM versus the TAM alone arm. And this is the shot we want you to take home, and that is on your left side is disease-free survival, on your right side is overall survival by recurrence score. And what you can see here is that it is prognostic, but it is also predictive. You can't see that from this slide. I took out the predictive side, I'm sorry. But that led us to wonder if that was true, is whether or not patients who have a low recurrence score, even though they have a relatively high risk of recurrence over the next 10 years because they're node positives, might not benefit from chemotherapy. And based on that, we designed a trial. Ana Maria Gonzalez from MD Anderson is the study chair of this, in which patients who have positive nodes and are hormone receptor positive and HER2 negative, if they wish to participate, have their tissue sent off for the 21 gene recurrence score. And if their recurrence score is less than 25 and they agree, they are then randomly assigned to receive chemotherapy or not. They all get appropriate endocrine therapy. That study's been open now for I think about 2 years. It's accruing faster than we thought it would, so we're quite pleased we should have an answer to that. In addition, we're collecting tissue from all of those patients, and so we'll be able to look forward. Furthermore, we've now gone back to 8814, and with Genomics Health, Inc., the same company that developed the 21 gene recurrence score, we now have permission to do what Jimmy was talking about with whole transcriptomic analysis, so that we can do both quantitative transcriptomics of all 15,000 or so genes as well as sequencing. And Kathy Albane's working hard on that, and I think we now have everything ready to go and should hopefully have data in the next 6 months. The second thing we've been working on are circulating tumor cells, and these are data in a study done before a pre-SWOG study, if you will. Massimo Cristofanelli led this, and we published in the New England Journal. Many of you have seen this. These are metastatic breast cancer patients who still had elevated circulating tumor cells at first follow-up. And what we found, if that's true, they had a very rapid chance of progressing over the next few weeks, frankly speaking, whereas if either they started without elevated CTCs or they converted from elevated to high, they have a pretty good prognosis. But not everybody had a bad prognosis, and that led us to wonder whether or not an early change at first follow-up might make a difference for these patients, or perhaps not. So led by Jeff Smirosh from our institution, SWOG started SO500 about 5 or 6 years ago, or more than that, I guess 8 years ago. We screened about 550 patients as expected. About half of them had elevated cells at baseline. As not expected, more of them responded to first-line chemotherapy than we expected. However, we still were able to then, if they still had elevated cells at first follow-up, they were randomly assigned to either staying on that therapy, a standard of care, or switching to an alternative chemotherapy. The good news is that was fully accrued about 1.5 years ago, and the DSMC is meeting today, and we're holding our breath and hoping we get data released to us. I can't tell you if we will or not, but we're hoping we will. A future study, we're going to look at CTCs led by George Somlo, and we'll be talking more about this at the breast meeting tomorrow, so I encourage you all to come. It's a real collaboration between my own lab and that of Peter Kuhn from California and James Hicks from Cold Springs Harbor, in which ER-positive breast cancer patients who've had no prior chemotherapy for their metastasis will be randomly assigned to fulvestrant plus everolimus based on the Bolero data, and fulvestrant plus everolimus and anastrozole based on Rita Mehta's study from SWOG suggesting that fulvestrant plus anastrozole is better than anastrozole alone. My laboratory will be doing something we call the CTC Endocrine Therapy Index, where we can measure ER, BCL2, HER2, and Ki67 in a quantitative fashion, as well as counting the cells. And we believe we may be able to identify patients who, although their primary tumor was ER positive, will behave as if they're ER negative and would have been better going straight on to chemotherapy. And Drs. Kuhn and Hicks will be measuring CTCs using a different platform, the so-called EPIC platform, generated at Scripps, and they'll be doing next-gen sequencing actually on individual cells using technology developed by Dr. Hicks. Very exciting collaboration. And again, if you want to hear more about this, come to the breast committee meeting tomorrow morning. Finally, we've had a great deal of interest in pharmacogenomics so we can see if we can identify inherited germline SNPs that affect drug activity or toxicity. This has been principally led by Christine Ambrosoni from Roswell Park in our group. This is one of our first studies suggesting that perhaps patients who have a SNP, excuse me, an MPO that doesn't permit deactivation or doesn't permit activation of cyclophosphamide might do worse than patients who have wild type, whereas if they were untreated, did not receive chemotherapy, they all did the same. A second area of interest is in hormone therapy, and again, using SO226, the trial by Rita Mehta, Susan Kadlebar from the University of Arkansas prospectively collected germline DNA, and those analyses are now finished, and we hope to have some data for you in the next few months. Bill Barlow is working on the analysis of these now. Finally, Christine has led a trial in SO221. Sorry, I have the wrong number there, not SO201, it's SO221. A trial led by Tom Budd of dose-dense versus dose-denser chemotherapy, if you will. As many of you know, roughly 10% of patients who get a taxane have life-altering neuropathy that's often permanent, and the question is why. So Christine and her group, taking the germline DNA that we collected, did candidate gene analysis for the Fanconi gene as well as a couple of others because of the fact that it's critical for repair of DNA damage, and she hypothesized that perhaps SNPs in Fanconi or BRCA1 would be related to neurotoxicity. And although I can't tell you how this slide works because I don't fully understand haplotypes, the fact is that it does look like this is the case. Either fortunately or unfortunately, in two other groups, CLGB and ECOG, similar efforts were made with other studies of taxanes. Similar hits were made, but in completely different genes. So we now have 3 different groups with 3 different hits for the same gene, same endpoint. And surprisingly, but very reassuringly to me, all 3 of these investigators, Brian Schneider, Deanna Krutz, and Christine, have agreed to play well in the sandbox, and we're hoping to pool all these data together and do a giant GWAS and see if we can actually validate which, if any, of these genes really does predict for this really severe toxicity, and perhaps then we can write future clinical trials to either avoid taxane in these patients or perhaps modify it with therapies. So right now, I think the results have been good for publication. They're sort of disappointing in terms of we really haven't found anything that is enough to justify a prospective validation trial for the chemotherapy issue, but I think the taxane approach is quite exciting. We have a couple of new initiatives I don't have time to go through. Zena Nale ran a neoadjuvant trial of bevacizumab and chemotherapy, and Lajos Pustai will be doing genomic profile of biopsies before and after chemotherapy. That's been sent to the federal government for review. Unfortunately, we don't have a federal government, but when we do, hopefully Lajos will be off to the races. And in S9313, an older trial of adjuvant AC versus adjuvant AC, don't ask. Yes, we did do that trial. In the old days, we thought that sequential single chemotherapy would be better than combination chemotherapy. Turns out the 2 arms are identical, but we have tissues on all these, and Priyanka Sharma has done a great job of leading a number of investigators to study BRCA-ness, not unlike P53-ness, in the triple negative cohort for that. That's also been sent to the federal government for review, and we're also waiting for the federal government to reorganize. So having said that, that's a brief overview of what we're doing. There are actually several other studies we've got in mind. We're more than interested in having anybody who wants to join us, and thank you very much for the chance to present.